They survived open space, no suit, no atmosphere, no protection, alive. The 14th of September, 2007, Baikonur Cosmodrome, Kazakhstan, pre-dawn. Floodlights cut through cold mist as a Soyuz rocket carried something extraordinary into orbit, something so small it was nearly invisible to the naked eye. The payload included living animals, bolted to the outside of the spacecraft, deliberately exposed to everything space could throw at them. The animals were tardigrades, Ramajotius varionatus times and times Rictursius coronafer, 8-legged, barrel-bodied, half a millimeter long, smaller than a grain of salt. And according to every textbook written before September 2007, every animal on Earth required one thing to survive: a barrier between its cells and the void. These had none. For 12 days they rode the outside of the Photon M3 spacecraft in a facility called Biopen 6, open vacuum, temperature swings of hundreds of degrees, cosmic radiation, solar ultraviolet, the full unfiltered spectrum that Earth's atmosphere normally blocks completely, the kind of UV that shreds DNA like paper. Ingemar Jönsson, a biologist at Kristianstad University in Sweden, designed the experiment. He had already spent years studying tardigrade resilience. He knew they were extraordinary. He did not know they were times extraordinary. The 26th of September 2007, the capsule returned. Jönsson's team recovered the specimens. They rehydrated the samples, a single drop of water, onto a dry biological crust, and waited. Some moved. That should not have happened. That single word, moved, changed the question biology had been asking about the limits of animal life. In 2008, Jönsson published the results in Times Current Biology. The numbers in that paper are still difficult to read without stopping. Specific numbers, black ink on white paper. Survival rates that should read 0. By the end of this video, the molecular mechanism that kept those animals alive will be named, and the one step in that mechanism biology still cannot explain will be laid bare. The textbook answer comes fast, too fast. Cryptobiosis. That is the word every biology professor reaches for. The idea is elegant. Tardigrades, when threatened, shed almost all their body water, fold inward, and enter a state so close to death that damage simply cannot find purchase. Dormant tissue, the reasoning goes, is inert tissue, and inert things are hard to destroy. It sounds complete. It feels complete. For decades it was complete. Then Ingemar Jönsson and his team at Kristianstad University sat down with the Photon M3 data, 12 days in low Earth orbit, September 2007, and the numbers stopped cooperating. The vacuum results held. Most animals survived exposure to the hard vacuum of open space. That part matched the model. Dormancy as shield, protection through stillness. The textbook confirmed. But then came the UV groups. Specimens exposed to unfiltered solar radiation, the full spectrum, including UVC, the wavelength that shreds molecular bonds, showed something the dormancy model had no answer for. Survival rates dropped sharply, not slightly, sharply. And in the animals that did survive full UV exposure, Jönsson's team found measurable DNA damage, strand breaks, real structural injury, recorded in cells that were supposedly locked down and protected. Jönsson published the finding in Current Biology in September 2008. His own words, the majority of animals survived exposure to space vacuum, whereas only a few animals survived the full spectrum of solar UV radiation. Read that twice. Vacuum, most survived. Full UV, almost none. If cryptobiosis were simply deep dormancy, if it were just protective stillness, the radiation damage pattern makes no sense. Dormant tissue does not selectively resist vacuum while remaining vulnerable to photon bombardment. The mechanism has to be more specific than that, more structural, something operating at a level the dormancy model never described. Dormancy explains the survival, but it does not explain the damage. That gap, that precise, measurable gap in the data, points directly at something inside the tardigrade that science, in 2008, had no name for yet. 9 years after those tardigrades came back from space, a team at the University of North Carolina sat down with the question nobody had answered. Not what survived, how? Thomas Boothby and Bob Goldstein published their findings in Molecular Cell in March 2017. The paper identified a class of proteins found nowhere else in nature, not in bacteria, not in plants, not in any other animal on Earth, only in tardigrades. They named them CAHS proteins, cytoplasmic abundant heat soluble proteins. The name is clinical, what they do is not. When a tardigrade loses water, when the last molecule of moisture leaves the cell, these proteins do not simply slow down. They transform, they vitrify, they become glass. A biological glass wraps around the DNA strand, physically immobilizing the double helix inside a non-crystalline, amorphous solid. Boothby's paper described it non-crystalline, amorphous, kinetically trapped solids that encapsulate and protect cellular components. Not dormancy, not slow down, structural transformation at the molecular level. Remember the promise from the beginning of this video, that by the end, the mechanism would have a name. Here it is, glass. The animal becomes glass from the inside. That is how the vacuum of open space failed to shatter the DNA. The glass held it still. Radiation passed through a structure that could not be further broken. But here is where the story refuses to close cleanly. When water returns, when that first droplet contacts the ton, the glass dissolves. The protein unfolds back into function. The animal wakes. And no one has characterized how. The rehydration step remains uncharacterized in the scientific literature. The mechanism that brings the animal back is the part biology still cannot describe. A creature smaller than a grain of sand solved a problem that has defeated every engineer who has ever tried to protect biological material in a vacuum. It solved it with a protein that builds itself into glass and then, somehow, forgets how to be glass. That answer is sitting in a drawer somewhere waiting. If you found this fascinating, subscribe. We investigate strange, real creatures like this every single day.